Good day, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, Eurofins Pharma Discovery Services, I'd like to welcome you to Recent Advances in Transporter Testing Strategies Enable Effective Identification of Potential Drug-Drug Interactions. My name is Elizabeth Lamb, and I'm the host and the moderator for today's event. I'd like to introduce our presenter for today. He is Kevin Kennedy, PhD, Principal Scientist, and Director of ADME-PK for Eurofins Pharma Discovery Services. Welcome, Dr. Kennedy. The presenter ball is yours.
Thank you, Elizabeth. Hello, everyone, and thank you for attending our webinar today. My name is Kevin Kennedy, Principal Scientist for ADME at Eurofins Pharma Discovery Services. It's my pleasure to speak to you today about our topic, strategies for in vitro assessment of transporter-mediated drug-drug interactions. I hope that this webinar will serve as a useful guide to gaining familiarity with the importance of assessing DDIs for metabolizing enzymes and transporter proteins in drug development. I also hope that the audience will leave with a good understanding of the current technologies that are used for the assessment of transporter interactions in vitro. For the agenda for today, we will introduce the topic of drug-drug interactions by first looking at some clinical examples and then discuss the relevance of predicting clinical DDIs from in vitro data. We'll examine the proteins which play a role in mediating these DDIs, both metabolic enzymes and particularly DDIs mediated through transporter protein interactions. And we'll also get a sense of regulatory perspective on how interaction of new drug candidates with these proteins should be assessed. We'll look in some depth at the strategies available for in vitro transporter interaction studies and the advantages and limitations of each. Finally, we'll discuss the implications of these in vitro DDI assessments for a drug development program and the capabilities that Eurofins has for performing these assays. So to introduce our topic, let's first define what we mean when we refer to a drug-drug interaction. Simply put, a drug-drug interaction, or DDI, occurs when the administration of one drug affects the pharmacokinetics of another co-administered drug or drugs. DDIs can result in increased or decreased exposure of the victim drug, resulting in adverse effects or decreased efficacy, respectively. Now, it's easy to understand that the risk of a drug-drug interaction increases with the number of drugs that are available to interact, and this can certainly be a concern in certain patient populations. Here is some data from a recent study highlighting the fact that many patients, particularly in the elderly population, are taking multiple medications simultaneously. And the multiplicity of these drugs increases the odds that one of these drugs will impact the PK profile of another. Our key concept for the discussion today is that in order to be able to predict potential DDIs prior to clinical studies, it's necessary to do comprehensive in vitro DDI profiling of a drug candidate. This involves quantifying the contribution of metabolic enzymes and transporter proteins to the drug's disposition. I think it's widely appreciated the role that metabolic enzymes such as cytochrome P450s or phase 2 enzymes such as UGTs have in drug disposition. But as we will examine today, in the last decade, it has become more apparent the important role that transporter proteins play, not just in the absorption of drugs, but also in how drugs are distributed and cleared from circulation. So in vitro transporter assays will be a particular focus here today. Let's start first with looking at a clinical example of a drug-drug interaction mediated through an interaction with a metabolic enzyme, in this case via inhibition of a cytochrome P450 enzyme CYP1A2. The example shown here is the NSAID rofecoxib, or Vioxx, which was identified in development as a time-dependent inhibitor of CYP1A2. In the clinical setting, when rofecoxib is administered together with a CYP1A2 substrate, the drug tizanidine, with the inhibition of 1A2 metabolic activity, plasma levels of tizanidine were found to increase approximately 14-fold. The plot on the right shows concentration versus time profile of tizanidine with and without rofecoxib, and you can see in the black circle plot the significant increase in the AUC when rofecoxib is co-administered with tizanidine. So clearly we see that interactions with drug metabolizing enzymes can have a relevant clinical impact, and the example shown here is one of many that have been documented. This is why assessment of compounds as substrates and as reversible or irreversible inhibitors of CYP enzymes is a standard part of early drug development programs. And regulatory agencies such as the FDA and EMA have issued guidance documents outlining recommendations for how these in vitro assays should be performed and how data obtained should be used to predict potential DDIs in vivo. In vitro CYP inhibition assays are typically performed with liver microsomes, which is the protein fraction of liver cells enriched with phase 1 metabolic enzymes, or with purified recombinant CYP enzymes. These assays can be run using drug probe substrates or fluorescent substrates. And shown here are some IC50 curves generated for CYP3A4 using the reference inhibitor ketoconazole with several different 3A4 substrates. This slide shows another example of a clinical DDI mediated through a metabolic enzyme, in this case through induction of a cytochrome P450 and induction of CYP3A4. The drug ibrutinib is an orally active cancer therapy, which is a substrate of CYP3A4. And in the clinical setting, when this drug is administered with a potent 3A4 inducer, rifampin, the increased CYP3A4 levels result in increased metabolism of ibrutinib and a corresponding decrease in the AUC by a factor of 10 and the Cmax by a factor of 13. And on the right, you see the dramatic decrease in the AUC profile when rifampin is co-administered with this drug. So since drugs may not only be inhibitors or substrates of CYP enzymes, but also inducers, in vitro assays to assess the potential for compounds to induce CYP production are also a common part of drug development programs. These assays are usually performed using messenger RNA levels as an indicator of CYP induction, or by measuring CYP enzymatic activity. Quantitative proteomics approaches have also been used to directly quantify levels of cytochrome P450s and transporter proteins. This slide shows some in vitro results generated internally in our lab in our high throughput 384 well CYP induction assay. This assay is performed with human cryopreserved hepatocytes and using messenger RNA levels to quantify fold induction of the CYP enzymes. The data shown here shows the increase in fold induction observable over time for the major inducible CYPs 3A4, 2B6, and 1A2 with the known inducers rifampin, phenobarbital, and omeprazole, respectively. Since the most common mechanism for CYP induction is transcriptional gene activation, another approach to gaining insight into induction of CYP proteins is to look at nuclear receptor activation. The PXR, CAR, and AHR nuclear receptors play a pivotal role in the expression of drug metabolism genes, which determine the clearance of xenobiotics. And we can look at nuclear receptor activation using a reporter cell system in which the activation of the nuclear receptor is functionally linked to a luciferase gene, and the activation can then be read out through fluorometric detection. This approach can be used to assess agonist or antagonist response on human nuclear receptors and nuclear receptors from other species as well. In summary, assessing in vitro CYP interactions early in development is necessary to understand and predict potential metabolism-based DDIs. A complete in vitro portfolio for CYP interactions will include assessment of a drug candidate as a substrate, as an inhibitor, and as an inducer of the CYP proteins which are relevant for DDIs. For the rest of the presentation, however, I'd like to focus our attention on the role that drug transporter interactions play in drug disposition and DDIs and approaches for assessing these interactions in vitro. Transporter proteins play a key role in the distribution and disposition of endogenous compounds and xenobiotics, and they are found throughout the body and in many organs. And in fact, there are over 400 membrane-bound drug transporters that have been identified. However, not all of these transporters impact drug disposition, and a great deal of research in this area has established that there are two main transporter families which play a role in drug disposition and are relevant with regard to clinical DDIs. One of these families is the ABC or ATP binding cassette family, which function by using ATP to provide active transport against the concentration gradient. These transporters are often referred to as efflux transporters, and this family includes the PGP transporter, also referred to as MDR1, the BCRP, MRP2, and BSEP transporters. The other relevant transporter family is the SLC or solute carrier transporter family, which function through facilitated ion-coupled or exchange transport. Transporters of relevance in this family include the organic anion transport, polypeptide transporters OATP1B1 and 1B3, organic anion transporters O2 and O3, organic cation transporters OCT1 and OCT2, and the mate transporters and ASBT and NTCP transporters. Now, the role of these transporters in disposition and DDIs is an active research area, and there is a continuing assessment and evaluation of which transporters are most important with regard to mediating clinically relevant DDIs. And at this point, I'd like to show some data which highlights the prevalence of transporter involvement in DDIs. This is data compiled from a recent survey from a group at the University of Washington Drug Interaction Database, where they reviewed in vivo PK data and in vitro data for 103 new molecular entities that were approved over the time period 2013 to 2016. The results from the survey are very striking in terms of how many clinically observed DDIs are related to transporter proteins. Of the 103 NMEs approved, 47 were found to be in vitro substrates of PGP, 20 were found to be substrates of BCRP, and another 8 were found to be substrates of the organic anion polypeptide transporters OATP1B1 and OATP1B3. In addition, transporters played a role in 46% of clinically relevant DDIs related to inhibition, although most of these were weak to moderate interaction. So a couple of points can be made here. First, we can appreciate why there has been a great deal of industry and regulatory focus in the last decade on the role of transporters in DDIs because of this prevalence. But another point I want to make is that even approved drugs can exhibit drug-drug interactions. And the idea behind assessment of in vitro CYP and transporter protein interactions is not to preclude a drug from further development, but rather to gather sufficient information to adequately predict and to assess the potential for a DDI so that if and when a drug is approved, it can be dosed safely and effectively. Let's look now at the distribution of these relevant transporters in the 4 key organs important for drug disposition: the intestine, in the liver, in the kidney, and at the blood-brain barrier. In the intestine, a number of transporters are expressed on the basolateral side of intestinal epithelia, but the ones of most relevance here are the efflux transporters PGP and BCRP. These transporters play an important role in limiting the absorbance and bioavailability of many drugs through active export out of intestinal cells into the intestinal lumen. In liver cells, a number of organic anion and cation transporters are localized on the plasma membrane of liver cells. And these transporters, particularly as we just saw on the previous slide, the OATP1B1 and 1B3 transporters, are important for the active uptake of drugs into liver cells. Hepatocytes also have another level of cellular architecture and form bile canaliculi. And efflux transporters active in bile canaliculi play a role in active secretion of endogenous compounds and drugs into the bile duct. In kidney cells, there are several organic cation and anion transporters in the basolateral membrane, which play a role in the active uptake of drugs into kidney cells, particularly the organic anion and organic cation transporters O1, O3, and OCT2. And there are also transporters localized in the apical membrane, which actively export drug compounds into urine. The PGP and the mate transporters are particularly relevant here. And then finally, at the blood-brain barrier, similar to in the intestine, the efflux transporters PGP and BCRP play a major role in active efflux and in limiting the ability of many compounds to cross the blood-brain barrier into the CNS. We saw earlier some clinical examples of metabolic-mediated DDIs, and here's an example of a clinical DDI mediated through transporter proteins. This is the example of the drug tenofovir, which is a drug eliminated unchanged in the urine by a combination of glomerular filtration and proximal tubular secretion. Clearance of tenofovir in the kidney is mediated through the involvement of transporter proteins. 20 to 30% of the drug is actively taken up by kidney cells via the OAT transporters and subsequently secreted from kidney cells into urine by MRP transporters in the apical membrane. Also, accumulation of this drug in kidney cells can cause nephrotoxicity. A number of drugs interact with these transporters and when co-administered can impact the clearance and nephrotoxicity related to tenofovir. For example, probenecid, which is an OAT inhibitor, when co-administered can block the uptake of tenofovir into kidney cells, which mitigates toxicity. On the other hand, when the drug ritonavir, which interacts with multiple efflux transporters, including MRP, is co-administered, secretion of tenofovir out of kidney cells decreases, and tenofovir levels accumulate in the cells with subsequent nephrotoxicity observed. So other examples of clinically relevant DDIs mediated through transporter proteins have been documented, but only relatively recently has an appreciation grown for the significance of the role that these transporters play in drug disposition and DDIs. And here we see a timeline which highlights that. And you can see in the last decade or so, regulatory agencies and the International Transporter Consortium have issued a number of publications and guidance documents on this topic. The concern for these groups and in the content of these guidance documents is in identifying which transporter proteins have the most likelihood of being involved in clinical DDIs, and with assessing how a new drug candidate interacts with these transporters in vitro studies prior to reaching the clinic. This is still an evolving area, and as research continues to shed light on the interplay of transporters and metabolic enzymes in drug disposition, opinion on what in vitro assays to perform and how to perform them is fluid. However, we can look specifically at the example of one current regulatory perspective on this topic, this excerpt coming from the most recent FDA guidance on drug-drug interaction studies. And with regard to their suggestions in terms of transporter assessment, because as we saw the significant role that PGP and BCRP play in a number of different tissues, the FDA currently recommends that all investigational new drugs be assessed as substrates of the efflux transporters PGP and BCRP. The FDA does have recommendations for substrate assessment on some other transporters as well, but this is dependent upon the expected clearance pathway for the drug of interest. For drugs where hepatic clearance is expected to be significant, the FDA recommends the drug be assessed as a substrate of OATP1B1 and OATP1B3. For drugs where renal clearance is expected to be significant, the FDA recommends the new drug candidate be assessed as a substrate of OAT1, OAT3, and the organic cation transporter ACT2. And then because there are clinically significant interactions demonstrated for a number of transporters with different drug classes, the FDA also recommends that all investigational new drugs be assessed as inhibitors of these transporters as well. In light of these recommendations, a comprehensive approach to in vitro profiling of transporters typically involves assessment of a new drug candidate as a substrate and inhibitor of the diversity of ABC and SLC transporters that may be involved in disposition. Not all of these may be specifically suggested in guidance documents, but as I mentioned, this is an evolving area and there is no complete consensus about which transporters are relevant. The BSEP transporter, for example, is recommended for inhibition testing by the European Medical Agency, but not currently by the FDA. Also, it is hard to predict which transporters may mediate the disposition of a specific drug, and in general, the more complete the transporter interaction profile of a drug, the better one is able to predict potential clinical DDIs. In terms of assay format, we will look in more detail at these approaches shortly, but in general, in vitro interaction studies are performed in whole cell models using cell lines which naturally express a transporter, or in cell lines transfected to overexpress the transporter of interest. Assays can also be run in membrane vesicles prepared from these transfected cell lines. A common approach for these interaction studies is to run these assays in a single concentration mode as an initial screen, and then if hits are observed, a follow-up multiple concentration assay can be performed in order to calculate an IC50. And depending on the substrate used to measure the transporter activity, readouts for these assays can involve fluorometric, radiometric, or LCMS detection. And finally, in addition to the recommendations as to which transporters should be assessed, regulatory guidance documents also outline suggested probe substrates, inhibitors, and parameters to be used for these in vitro assays as well. So now that we've looked at the overall approach to in vitro transporter assessment, what I'd like to do now is to look more in depth at the different assay formats and the technical aspects of how these assays are performed. Keep in mind that the goal of these in vitro assays is to obtain data which can predict an in vivo outcome. So what we'll see is that the models for in vitro transporter assessment involve a trade-off in being able to look specifically at the role of individual transporters versus models which attempt to recapitulate the complexity of all transporters and metabolic enzymes and the interaction of these proteins in the disposition of a drug in vivo. But let's first start by looking at a relatively simple system for assessment, the strategy of vesicular transport assays. This approach utilizes membrane vesicle preparations from cells which have been transfected to express specific transporters. And this is a convenient way to assess drug candidates as either inhibitors or substrates of particularly the efflux transporters. So in the schematic, we see the inside-out vesicle expressing the efflux transporter, and with a supply of ATP, the transporter functions to actively uptake the substrate of interest into the vesicle. Drug candidates can be added to assess the ability to inhibit that activity, or they can be added directly to be tested as substrates to see if the compound is internalized in the vesicle through active transport through the transporter. Vesicles are also a convenient way to look at species difference in efflux transport, as membrane vesicles prepared from cells expressing rodent proteins are commercially available. A limitation of this approach is that compounds with medium to high passive permeability are difficult to test, as number one, they pass easily through the membrane vesicle in the absence of active transport, and number two, are not retained in the vesicles and tend to leak out. And so it's difficult to identify the contribution of the transporter for compounds of this type. Also, again, this is a simple model which allows for assessment of individual, primarily efflux transporter interactions. And this is not a model which can provide insight into the contribution of multiple transporters. And since there are no metabolic enzymes in these vesicles, the model doesn't allow for the assessment of the interaction of transporter and metabolic enzymes on drug disposition. Here's an example of some inhibition data generated in a membrane vesicle model, in this case measuring inhibition of the bile salt export pump, or BSEP transporter. This data was obtained using membrane vesicles prepared from human embryonic kidney cells transfected to overexpress the BSEP transporter. And the assay measures transport of a radiolabeled bile acid into the vesicle. The table shows some IC50s generated for various test drugs and the IC50 plot for cyclosporin, a known BSEP inhibitor. Now, in order to examine the contribution of multiple efflux transporters in drug absorption, a widely used strategy has been the use of cellular permeability assays in a transwell format. The cell lines commonly used for this purpose include Caco-2, which is a human adenocarcinoma cell line, MDCK2, which is a canine kidney cell line, and LLC-PK1, which is a porcine kidney cell line. We find that the Caco-2 model in particular is very useful for assessing drug compounds as inhibitors or substrates of the efflux transporters PGP, BCRP, and MRP2. And in this approach, the cells are grown on a porous membrane in a transwell plate. And in this format, they will grow to their morphology, in which they form a monolayer with tight junctions between the cells. And in this format, we can then add the compound. A test compound can then be added to either the apical or basolateral side of the monolayer, and the ability of the compound to pass through the Caco monolayer in either direction, either by passive diffusion or by active transport, can be assessed. An advantage of the Caco cellular permeability model is that the cells express all of the transporters relevant for absorption. And permeability results in this model do correlate well with in vivo absorption and bioavailability, provided the monolayer is well characterized and forms tight junction. These assays can also be run in a 96-well format and are amenable for high throughput screening. A limitation of these assays is that the focus here is on intestinal transport, although because of the involvement of PGP and BCRP in both the intestine and the blood-brain barrier, the Caco model is often used as a surrogate model to assess the ability of compounds to cross the BBB. Another limitation here is that there can be species-specific difference in transporter expression and activity, and results obtained in permeability assays in these human, dog, and porcine cell lines, the cell lines commonly used in this approach, may not always correlate. This slide shows data obtained using the Caco-2 cell line in a transwell permeability assay to assess a test compound as a BCRP substrate. For substrate assessment, the permeability of the test compound is determined in both A to B and B to A directions, with and without the presence of a specific transporter inhibitor. In this case, we are looking at the drug estrone sulfate, which is a BCRP substrate. And the plot on the left shows that in the absence of inhibitor, the permeability of estrone sulfate in the B to A direction is much higher than in the A to B direction, indicating that there is active efflux taking place. However, when a specific BCRP inhibitor, KO-143, is added, the permeability in the A to B and B to A directions are closer to each other, and the B to A to B efflux ratio, which in the absence of inhibitor is above 10, now decreases to a value close to 1. So again, this is a nice model for looking at the activity of these efflux transporters, provided one has a specific inhibitor for the transporter of interest. An extension of the transwell approach using these cell lines has been the creation of cell lines genetically engineered to knock out expression of efflux transporters. These cell lines can be used in permeability assays to identify transporter-specific substrates and to gain insight into complex interactions where a drug may be a substrate of multiple efflux transporters. In this graph, we see permeability data obtained in a subclone of the Caco cell line in wild-type cells and in cells where individual transporter activity has been knocked out. The bars shown here are the B to A to B efflux ratios for a number of different transporter substrates. And if we look, for example, at the green bar, this is the compound estrone sulfate, which we just saw in the previous slide as a BCRP substrate. And similar in the previous slide, we see that in the wild-type cell line, there is a high B to A to B efflux ratio, but in the BCRP knockout cell line, the ratio decreases close to a value of 1, and which is not the case in either the MDR1 or MRP2 knockout. So this again confirms the fact that BCRP is involved in the active efflux of this drug. Cellular permeability assays are very useful models for assessing efflux transporters and intestinal absorption. But in order to look at a broader series of transporters, the most commonly used approach are cell-based assays using cell lines transfected to overexpress specific transporters. Cell lines used for this purpose, such as human embryonic kidney cells, Chinese hamster ovary cells, and canine kidney cells, do not express human transporters, so they are a good blank canvas, so to speak, for overexpression of these proteins. These types of cellular models are very useful for testing compounds as substrates and inhibitors of both ABC and SLC transporters, and also are good models for transporter kinetic studies. A limitation here is that these are cellular models, which again look at the interaction of a drug with one transporter, or potentially if one has a doubly transfected cell line, possibly 2 transporters at a time. And also these cells do not have metabolic competency, so these transfected cell models are not useful for examining the involvement of multiple protein interactions on drug disposition. A more complex cellular model, which does allow one to assess the interaction of multiple transporters and metabolic enzymes, particularly for hepatobiliary transport, are liver cells which are grown in sandwich culture. Hepatocytes which are grown in this fashion between layers of collagen develop morphology which is similar to in vivo hepatocytes, in that they express drug metabolizing enzymes, they have functional uptake and efflux transporters, and they also develop bile pockets which are similar to the bile canaliculi found in vivo hepatocytes. This model allows one to obtain a more complete picture of all the proteins involved in the disposition of a drug, including interactions of metabolic and transporter proteins. However, given the complexity of interactions which are possible in this type of model, assessing the interaction or contribution of a specific transporter with a drug may be difficult if specific inhibitors are not available, and this can often be the case. As such, a further development of this approach has been the development of engineered hepatocyte cell lines where expression of specific transporters has been knocked out. And as we saw with knockout Caco cell line, this allows one to isolate the role of individual transporters in complex interactions where specific inhibitors may not be available. And there are even more complex models which have been used for assessment of transporters in disposition in DDIs. For example, liver cells grown as 3D spheroids, or recent technology attempting to create organ-on-a-chip models. Transporter assays can be run in tissue slices or in whole organs using a perfusion approach. And then finally, moving beyond in vitro models, these types of studies can be done in whole animals. For example, using bile duct cannulated animals to explore hepatobiliary clearance, And there are also transgenic animal models that have been developed where transporter activity has been knocked out, or humanized models where the native animal transporter has been knocked out and the expression of the human transporter engineered back in. So now that we've looked at how these in vitro transporter interaction assays are performed, I'd like to conclude by discussing briefly the utility of this data in a drug development program and how these results are used. I often get questions from clients who have done these studies at our facility, and particularly when, as often happens, there is some interaction with a transporter or enzyme about what needs to be done next. Now again, because the goal at this point is to use this in vitro data to predict the likelihood of a clinically relevant effect, the in vitro data needs to be assessed in the context of the PK profile of the compound. If, for example, a drug has an in vitro IC50 on a transporter, which is well above the expected circulating concentration, the concern about a possible clinical DDI will be minimal. And various guidance documents have useful decision trees which are helpful in determining if additional clinical DDI studies are needed. This is an example of an ITC decision tree for the OHE1, OHE3, and OCT2 transporters. And as we saw earlier, interaction with these transporters may be clinically relevant for drugs primarily cleared via a renal pathway. So in the decision tree, there are some questions that if the answer is affirmative with regard to the expected renal clearance of the drug, and the drug is identified as an in vitro substrate of these transporters, it may be ultimately necessary to perform an in vivo DDI study with a sensitive inhibitor of these transporters to see if any significant interactions occur. This in vivo study does not need to be performed immediately after the in vitro assessment. There can be further development of the drug candidate, and if and when it reaches the clinic, the in vivo DDI study is performed so that the drug can be labeled appropriately and administered safely with other medications. So just to summarize what we discussed today, we saw that clinically relevant DDIs that are mediated through metabolic or transporter proteins are prevalent and should be assessed prior to regulatory submission. We saw that the role and number of transporter proteins in drug disposition in DDIs is complex, and opinion continues to evolve as to which are the most relevant. We looked at a variety of technologies and assays that are available for in vitro assessment of CYP enzymes and transporter proteins. And we saw that interactions with metabolic enzymes or transporters identified in vitro may necessitate eventually an in vivo clinical DDI study. All of the in vitro assays discussed in the webinar today can be performed at the EPDS Center of Excellence for ADME in St. Charles, Missouri. We have a broad portfolio of assays not only for drug-drug interaction assessments, but also for examination of solution properties, absorption, metabolism, and toxicity. Results from these assays are useful to guide decision-making early in the drug development timeline and can be used to prepare regulatory submissions for lead candidates. We can also provide custom services to support specific needs clients may have for their projects. And as a whole, EPDS has a broad portfolio of services in addition to ADME-Tox, covering in vitro pharmacology, chemistry, immunology, oncology, and safety testing. We have provided drug discovery and development services for over 40 years and can be a valuable single-source partner for clients across the drug development spectrum. And you can find more information about EPDS and our capabilities at our website. Finally, I'd just like to acknowledge some of the people that were involved in generating some of the data that you've seen here today, and also in helping with the content of the webinar. I'd like to thank the ADME and Tox team in St. Charles for all the hard work they do, and for the marketing group for their assistance in generating content for the webinar. And now I would be happy to address any questions from the audience on any of the content that I've talked about today. Thank you for your time.
Thank you very much, Dr. Kennedy. Yes, we have several questions. Our first question, you mentioned that many of the CYP450 and NR assays are available in different species. How similar are the drug metabolism pathways in rodents versus humans?
Yes, okay, thanks. That's a good question. So rodent models can be predictive for many aspects of human ADME, but in terms of drug metabolism, it is common to find differences in how rodents metabolize compounds versus human. That's why typically rodents are not commonly used to assess drug-drug interactions due to poor clinical translatability of the results in rodent to human. for example, midazolam is a human 3A4 specific probe, which undergoes transformation via CYP2C in mouse, and rifampin is a positive inducer for CYP3A in humans, but there's no induction of CYP3A4 by rifampin in rats.
Okay, so there are definitely some differences.
Yes, yes. So again, typically rodents are not commonly used to assess drug-drug interactions due to the poor translatability to the clinic, and that's why, as I discussed earlier, there are animal models where the native rodent or animal transporter has been knocked out, but the human transporter engineered back in.
Okay. A question from Graham. For CYP450 induction studies, do you use plated hepatocytes or sandwich culture? And why do you select the particular model you use?
So for our CYP induction studies, we use primary human hepatocytes, which are cryopreserved. We find that this is a good model for assessing CYP induction, and we typically assess CYP induction measuring messenger RNA levels, as this is the approach that is suggested by regulatory guidance documents. But we also have the capability of running CYP induction assays with measuring enzymatic activity. But yeah, I mean, cryopreserved hepatocytes are a relatively convenient and less expensive and less complex way to assess CYP induction in an in vitro model.
Thank you. And a question from Chandra. Do you have any experience using a solubilizing agent for low soluble compounds for transporter inhibition studies? So
yeah, we commonly don't use any specific additives in the assay to increase solubility, as the cells that we use for the assessment tend to be sensitive to surfactants or DMSO content that is in the assay mixture. Now, in terms of intestinal transport, looking at Caco permeability in a transwell format, we do commonly add bovine serum albumin to the incubation for compounds which tend to have low recovery and which tend to have non-specific absorption to the plate or to proteins. So that actually is a very effective way at improving solubility and recovery of compounds in the Caco transporter assessment model.
Okay. Our next question. If an in vitro drug transporter interaction is identified early in development, what are the next steps that should be taken?
So we saw in the final. I'll go to that slide real quick. So as I mentioned, the goal here is in these in vitro studies is to identify the transporter and metabolic enzyme interactions that could be relevant for disposition. But this data needs to be used to looked at in light of the expected PK profile of the compound, and that may require some pharmacokinetic modeling using physiological parameters. And again, the decision trees and guidance documents also have recommendations on how the in vitro data should be looked at in the context of PK data, with ultimately what you're trying to do is decide if the interaction is sufficient or is likely to result in a potential clinical DDI. And then ultimately, if that interaction is strong enough, you may require eventually doing a clinical DDI study for your drug with a sensitive substrate or inhibitor of the transporter or CYP enzyme that that drug interacts with. Now, in many cases, regulatory agencies will allow clinical DDI studies to be completed post-marketing. So again, this is not if an interaction is identified in vitro, that doesn't necessarily mean preclude the drug from further development. It just means that eventually you may have to do a clinical study to demonstrate potential a clinically relevant DDI.
Okay. Another attendee asks, how efficient are the fluorescent dyes for drug transporter studies?
The fluorescent dyes we are very effective. It's not so much the fluorescent dye itself that's the issue. The issue with using transfected cell lines for transporter assessment can be the level of expression that you get in the transfected cell, and if that level of expression tends to diminish over time as you culture that cell line. So that's really the more important issue when doing those fluorescent studies with transfected cells is trying to establish if you have sufficient level of expression of your transporter of interest.
All right, our next question. How different are guidelines proposed, transporter testing guidelines, from regulatory agencies around the world?
Yeah, so there is some difference of opinion in terms of the regulatory opinion about which transporters to look at, both as substrates and inhibitors. And for example, the BSEP transporter is one example where the EMA does recommend that a new drug candidate be assessed as a substrate, whereas the FDA recommends that it be considered, and whereas the regulatory guidance from Japan does not recommend that. And the mates are another example of transporters which there is differing opinion on from regulatory agencies. So yeah, as we discussed, these opinions are evolving, and each particular regulatory agency may have a different opinion as to which transporters are relevant. But the ones that we spoke of from the FDA are, in general, are also recommended for substrate inhibition testing by both the EMA and in Japan as well.
All right. And Graham has asked for a bit of clarification regarding slide 23. He understands that you use human cryohepatocytes, but he asks, do you culture them in a monolayer or in a sandwich layer like you showed on slide 23?
Yeah, so we currently don't grow the hepatocytes as sandwich culture for our standard SIP induction testing. It is a strategy that we can employ for particular custom projects, but for our standard assay, we do not use sandwich culture hepatocytes.
Okay, understood. Thank you. For hepatic and renal transport evaluations, do you conduct tests in the presence or absence of proteins in the media?
In our transporter assessment assays, we do not typically add any additional protein component to the cell mixture.
Okay. Well, we do have a couple more questions, but it is time to wrap up. So I'd like to say again to our presenter today, Dr. Kevin Kennedy, thank you so much for your presentation. I'd like to thank Eurofins Pharma Discovery Services for sponsoring today's web symposium today. And finally, most of all, I'd like to thank those of you who came and spent some time with us. We hope you found some answers that will make your research a bit more accurate going forward. So thank you all so much, and have a great day.